Welcome everyone. I am Raji here from NextGen Academy. So we are here for yet another interactive session on plant respiration, right? So today we are discussing a very important topic, that is electron transport chain, or which you also call it as ETC, right? So as you know that aerobic respiration is a respiration wherein maximum energy from a molecule can be harvested, that is around 38 ATP. So we have already discussed various steps contributing towards the aerobic respiration, right from glycolysis, link reaction and Krebs cycle. But all the way we are not coming across any process, or collectively they are not giving us 38 ATP. Like for example, if you talk about glycolysis, the net gain of ATP, direct ATP in glycolysis is only 2. In link reaction, there is no direct ATP. As well as in Krebs cycle, when we talk about, again, there is only 2 GTP. So out of 38, we have received only 4 ATP molecule. So where are the rest of the 34 ATP coming here? Out of 38. So for the rest of the 34 ATP molecule, one of the major process that is a part of aerobic respiration is what we are going to discuss today. That is our electron transport system. So what is happening during electron transport system that is giving an organism so much of energy, right? So we will first talk about what exactly is happening there, where it is happening, and what all complexes and the process that is involved during the electron transport system. So first we will talk about the process here, right? So electron transport system is the final step in aerobic respiration, right? So when I say it is a final step in aerobic respiration, so that means it only occurs in aerobic respiration. It is not a part of anaerobic respiration, right? So it is a final step in aerobic respiration to harvest maximum energy, right? So maximum energy of aerobic respiration is harvested from electron transport system. So what exactly is happening in the process which is what you say, releasing so much energy? Because as we know that there is no possibility of any extra ATPs being released here, right? So what is happening here? So all the while that we are we studied aerobic respiration, along with ATP, we also received a lot of reduced coenzyme. So here I used a word reduced coenzyme. So let us see what do we mean by reduced coenzyme. So here final step in aerobic respiration to harvest maximum energy, right? So to harvest maximum energy from what? From reduced coenzymes. So to harvest maximum energy from reduced coenzyme. See here I have used the word reduced coenzyme. So what do we mean by reduced coenzyme actually, right? So I will tell you here example of reduced coenzyme that is released during the process of aerobic respiration, that is NADH plus H. This is one example of reduced coenzyme. Another example is FADH2, right? So these two molecules, if you remember, lot of these molecules are being released during glycolysis, during link reaction, as well as during Krebs cycle. So what exactly is the use of these molecules? Definitely these are biological reducing agent, but other than being used as a biological reducing agent, these also are storing a lot of energy, or these can be used to generate ATP, right? So electron transport system is a process wherein ATP can be generated with the help of these reduced coenzyme, right? So now how this reduced coenzyme can actually release ATP, right? So in a very simple way, let me just try to tell you here. For example, if I talk about NADH, right, or NADH plus H, right? So here it is having this hydrogen, right? So this is which form of this coenzyme? If you remember, NAD, FAD. These are also called as coenzyme. We have already studied in the chapter called biomolecule. We have a concept there as cofactors, right? So cofactors are additional non-protein components which helps in the activation or in helping in improving the activity of the enzyme. So one of the example of this cofactor was coenzymes, right? And the example that is given in our textbook for coenzyme is NAD and FAD. So nicotinamide adenine dinucleotide and flavin adenine dinucleotide. There is one more example, FMN, that is flavin mononucleotide. So all these examples or all these components are known as coenzymes, right? So these coenzymes can be present in two forms. It can be present in oxidized form or it can be present in reduced form, right? See, all these metabolic reactions that is happening in the plants or in, the organism, right? So these are generally redox reaction, right? So unlike the reaction that we do in test tube where we have to add reducing agent or oxidizing agent, what about in cell? What kind of reducing and oxidizing agent is used? So these coenzyme, right? It is one way of forming the reducing and oxidizing agent. That is the reason we call them also as biological oxidizing agent and biological reducing agent, right? Like for example, if it is in this form, NADH plus H form, it is reduced form, right? So this form can be reoxidized back. That means it can be reconverted to NAD plus, right? So when I say NAD plus, it is which form? It is oxidized form. So this is a process that is happening in the cell. So when it is getting oxidized, what it is releasing as a part of this oxidation? It is releasing the hydrogen molecule, right? It is in a very generalized form, I am telling you. Now, this hydrogen molecule helps in ATP synthesis, right? So this hydrogen molecule helps in the synthesis of ATP. So how this hydrogen molecule help in the synthesis of ATP? Because when we talk about ATP synthesis here, always remember ATP is produced from ADP and inorganic phosphate. It helps in the formation of ATP, right? So now this process, although we call ATP as energy currency of the cell, to produce ATP also energy is required, right? So where is that energy coming from? That energy is donated by these hydrogen. See here, the hydrogen which is coming from oxidation of this reduced coenzyme helps in the release of energy. That energy or that proton energy is used to convert ADP into ATP, right? So that means these two processes are linked to each other, right? So that means NADH2 can help in the formation of ATP. Now this process what you are seeing here, using NADH2 or reoxidation of NADH2 helping in the formation of ATP, this phenomena or this process here is known as oxidative phosphorylation. So what do we call that as? We call it as oxidative phosphorylation, right? So as I was telling you, electron transport system or electron transport chain is a process wherein maximum amount of energy can be harvested from where the energy equivalent that is stored in NADH2 and FADH2 can be used to convert ADP to ATP, right? So the process by which ADP to ATP formation happens during electron transport system is called as oxidative phosphorylation. See here, two words are used here. You have oxidation, you have phosphorylation. Oxidation of reduced coenzymes, right? And phosphorylation of ADP to ATP, right? So oxidation of reduced coenzyme and phosphorylation of ADP to ATP. So this is a combined process. So the process by which electron transport system helps in the release of energy is known as oxidative phosphorylation, right? So let us just see here, right, a bit more about the phosphorylation process. So it will be easy for us to understand the phenomena in a more clearer form, right? So let's see, right? So when I say the word phosphorylation, right? Now, phosphorylation is a term simply means addition of phosphate group, right? So now with respect to respiration, when you talk about phosphorylation, it is a process where ADP can be converted to ATP, right? So now with respect to our respiration process, there are two types of phosphorylation, it seems, right? One is substrate level phosphorylation. The second one is called as oxidative phosphorylation, right? So what are the two types of phosphorylation we are having? Substrate level phosphorylation, oxidative phosphorylation, right? So there is one more thing here. Substrate level phosphorylation can also be abbreviated as SLP. So in many MCQs, you might have seen the term SLP, right? So the ATP that is produced by SLP process, right? So now here one more thing we should be keeping in mind. This is also known as direct ATP, right? So the ATP molecules that are produced as a result of substrate level phosphorylation is known as direct ATP, right? So what exactly do we mean by that? Substrate level phosphorylation, donor of phosphate group is a substrate. So any phosphorylated substrate can donate the phosphate group which is accepted by ADP and help in the formation of ATP, right? So that is what we call as substrate level phosphorylation, which we can also call it as direct ATP, that we should remember here, right? So now come to this side, right? So now this here, what I wrote, a process called as oxidative phosphorylation, right? So when we talk about oxidative phosphorylation, right, here the energy, it is also abbreviated as OP, right? This is also known as indirect ATP, right? So here ATP produced due to oxidation of substrate, right? So these are the basic differences between substrate level phosphorylation and oxidative phosphorylation. One thing we should always remember, or we should keep it in mind, that substrate level phosphorylation can occur in both aerobic respiration as well as anaerobic respiration, right? Whereas oxidative phosphorylation is only part of aerobic respiration. It never or it will not happen during anaerobic respiration. So this only can be seen during aerobic respiration, right? Now aerobic respiration in all organism, it is not like that ETC can only be happening in plants or animal because they have mitochondria, right? The organism which doesn't have mitochondria and they are aerobic, like for example bacteria. Bacteria are prokaryote. That means they don't have any membrane-bound cell organelle like mitochondria. But yet, if they are aerobic, how do they do this electron transport system or oxidative phosphorylation? In them, the electron transport system occurs in their plasma membrane, or what you call as a part of plasma membrane, which can also be called as mesosomes, right? So we are very familiar with that term. We have already used that term in the chapters like biological classification and cell the unit of life, right? So this is in general about your type of phosphorylation. So when we talk about respiration, right, the phosphorylation that is making much difference, which is generating high amount of ATP, is your oxidative phosphorylation. Similarly, when we talk about photosynthesis, right, if you recall, the type of phosphorylation in photosynthesis is called as photophosphorylation. What do we mean by photophosphorylation? The phosphorylation which is happening in the presence of sunlight. So in photosynthesis, ATP is produced in the presence of light energy. That is the reason we are using the term photophosphorylation. So if you recall in the chapter, it is also associated with ETC. So how they have given the topic of ATP generation in photosynthesis? That is cyclic, non-cyclic photophosphorylation and electron transport. That means always this electron transport is associated with the process of phosphorylation, that is generation of ATP, right? So whether it is respiration or photosynthesis, right, the process of ATP formation, the term that we use there is phosphorylation, right? So based on the process, it can be photophosphorylation or it can be oxidative phosphorylation, right? So this is a general idea of what exactly is happening during electron transport system that is helping in the generation of so much amount of so many molecules of ATP, right? So this is a generalized idea, right? So now we will just have a talk about where exactly the process is happening, right? So that means if I have to see what is the site of electron transport system, right? Or where exactly this electron transport system is happening, right? So ETS in eukaryotes, when we talk about in eukaryote, it is happening in inner mitochondrial membrane. So where it is happening in eukaryote, inner mitochondrial membrane, right? Whereas in prokaryote, as I told you, the site is plasma membrane, because they do not have mitochondria. So there is no question of them doing the process in mitochondria, right? But then we do have aerobic bacteria. So where do they do their aerobic respiration, etc? They will do in their own plasma membrane, a part of plasma membrane that you also call it as mesosomes, right? So this is about the site of electron transport system. So always, and again I am repeating, we should always keep it in mind, ETS happens only during aerobic respiration. It is not a part of anaerobic respiration. So this is all about the general idea of what exactly we mean by ETS and how it is beneficial in the process of aerobic respiration. As we have discussed, ETS help to generate maximum ATP. So when I say maximum ATP, right, how much ATPs are generated as a result of ETS? How much ATP is generated during ETS? Let us see here. When I say number of ATP, right? So number of ATP in aerobic respiration, right? So how many ATP is produced during aerobic respiration? So all of you are aware of the number, right? So the modified number is 38 ATP, right? In the previous textbooks, they have mentioned about 36, right? That is also not wrong. But when we talk about the, MCQ or the question when it comes into exam, right? So we always have to go for 38 because it has been corrected in the textbook, right? Why it is corrected? See here. 36 or 38 depends on what kind of cell we are talking about, right? So certain cells in certain organism produces 36 ATP, but in general, the number of ATP molecule, theoretically, when I say 38 ATP molecule, the theoretical value of ATP molecule that is generated during aerobic respiration is 38 ATP. That is the reason we have to go with this number. Which number? That is your 38 ATP, right? So now when we talk about 38 ATP molecule, right, in this case, 4 ATP molecule is said to be direct ATP, right? When I say direct ATP, what do I mean by that? Through substrate level phosphorylation, right? And that means how many is pending? 34 ATP, right? 34 ATP will be indirect ATP. Indirect ATP means through oxidative phosphorylation, right? What do we mean by again oxidative phosphorylation? It is a process of ATP generation that happens during electron transport system or electron transport chain process, right? So this is what we are going to study now, right? So how this 34 ATPs are generated, right? So a scheme of electron flow is given in our textbook. That picture is very important. It will explain us how the electron when it is transported, right, protons also are released, and how this proton is helping us in the formation of ATP. See here, exactly what is helping in the process of ATP generation is proton or H plus, right? If you recall the previous chapter, that is photosynthesis in plant, right? There also we had a concept wherein it is explained that proton energy can be utilized for ATP generation. So there they have given a term called as chemiosmosis, right? The same chemiosmosis is happening here also. So now so much of terms are there, like, so how it is possible? One way electron transport is happening, then we are talking about chemiosmosis, we are also talking about oxidative phosphorylation. So all these three words are not same, right? But all the three words or all the three process ultimately helps in the formation of what? How many ATP? 34 ATP molecules, which is a huge number. That means most of the ATP that is generated during aerobic respiration is coming from the process that we describe as oxidative phosphorylation, right? So just we'll have a general sketch on all these concept that just I said. That means, right, so when I say oxidative phosphorylation, right, oxidative phosphorylation will release electrons and protons, right? Now this protons forms proton motive force, right? PMF or proton motive force. We'll have an explanation of this when we see with the help of the picture that we have in our textbook. The proton motive force, nothing but proton energy, right? So because the oxidation of coenzyme is happening, it is releasing the protons. Huge amount of protons when they go from one end to another end, or from the higher concentration to lower concentration, or when they move through the proton gradient, it creates lot of energy. That energy is what we call as proton motive force, or what you call as proton energy. And this proton energy is the one which helps in ATP formation, right? So now here the process of ATP formation with the help of this proton energy, right? This is what you call as chemiosmosis. So chemiosmosis is an inherent part of any ATP generation, which is happening during phosphorylation, respiration or photosynthesis, if a proton movement is involved, right? Because proton movement creates this energy, and ATP formation with the help of the proton energy. The process is called as chemiosmosis, whether we are talking about photosynthesis or respiration. So the ultimate energy formation, ultimate conversion of ATP to ADP, right? So if I have to write it, in an equation form, ADP plus inorganic phosphate, it will give you ATP, right? Is guiding this process. This process is guiding by with the help of proton energy, right? So this is what I was talking about. This process which is happening here, this is what you call as chemiosmosis. So everything combined into one. We have oxidative phosphorylation here. we have electrons getting moved here. Ultimately, we are also having your chemiosmosis, right? So for this thing to happen in a cell, there are certain requirements, right? So what all things are required for the process, how the process happens in the cell, we will continue to learn in the next session. Thank you.